put in.
Okay, there you go. Yeah.
Yes, it's 1 o'clock.
Not really.
Yes. Oh, wait, does that work?
No.
No. Hopefully I have enough things. Hopefully I have things enough circled on there.
Yeah. I think I'm still working.
No, use the laser that's on there.
Is the one that's on there?
No, it's too finicky.
It used to be like all the rage. Everybody had like laser pointers in their pockets, but we don't really do that anymore.
I'm just giving it.
Used to like on Zoom saying like, and it's in the upper left. Like doing a really physical description of where things were. Yeah. No, that's weird. So it was like, used to be that like when you graduated, people gave you laser pointers. Now. You do like cool cat boy. I did not teach that. We're gonna get started?
Yes.
Yes, okay. All right. So thanks for coming today. It's a rainy day, so what better to do than talk about mushrooms, right? So Nadia is gonna talk to us today about the work that she's been doing this year as part of her senior thesis. And she's got some really cool stuff. So we're excited to hear it. And we'll take questions at the end. So thanks for
Yes.
joining us.
There will definitely be space for questions. If you brought anything to write anything down, that would be extremely helpful. So once again, I'm Nadia Small. This is my biochemistry senior thesis work that I did on the psychoactive constituents of the red mushrooms with the white spots, Amanita muscaria. So these mushrooms have been reported in ancient texts and in Viking texts. There's a lot of Siberian shamanism related to the use of these mushrooms in spiritualistic practices, as well as other Central Asian practices. It's believed that these mushrooms may be what's referred to as the soma in the Hinduistic texts. So there's a long history with these things, and they've started to make their way into Western medicine. And there's a lot of lack of knowledge about how these mushrooms work and how they're causing the subjective effects that they do cause, which we will get into. There's some folklore associated with the belief that their dissociative effects and their use by Central Asian shamans led to the imagery that we currently have of Santa Claus. So now as they've made it to the US, things have changed to where these are now a topic of fairy tales, but they're also moving their way into the pharmaceutical industry to look into what these mushrooms can be used for in treatments for various disorders. Currently, these mushrooms do grow out of the ground in North America. Mostly, they're going to be growing out of the ground in Asia and Europe as well, but they do grow out of the ground in North America, so you might run across one of these and you might pick it. They are toxic, but they're also sold in 49 states, and there's a lack of regulation on their sale. You can buy them, and they're still illegal to consume at that point, though. So what they're currently used for, they're referred to as nature's benzodiazepine. They have the same mechanism of action that alcohol and benzodiazepines have. They're used in tinctures for topical naturopathic pain relief, and they're still also used in spiritualistic practices and meditations. And in the pharmaceutical industry, there have been isolates that have been turned into drugs, but one isolate that has been used the entirety of the mushroom to make an insomnia drug called gaboxetal. That made it to phase 3 of clinical trials. It did fail at that point due to hypnotic side effects, but that did show that there is a possibility for progress in the pharmaceutical industry with these mushrooms. So what I did was I did a 3-step study where I looked at the effects of these mushrooms on neuronal development in a cell morphology study. I did a chemical analysis of just what I could pull out of the mushrooms to see exactly what those psychoactive compounds were and what some of their properties were. And then I looked to see how my experimentation that I did this semester could be revised and work as preliminary data to continue this experimentation in the future and to look at some more of the specific effects I was not yet able to address. So the first section is this cell study. So I wanted to look at how treatments with these mushrooms affect neuron development. So a very common way to look at neuron development is to look at neurite outgrowth. These are just any of those outgrowths that you see when you're looking at a neuron. You're seeing that structural change that looks like these cells are reaching out and communicating with each other. So abnormalities to these neurite growths do occur in disorders such as autism, schizophrenia, and epilepsy, and there are a handful of drugs that have been shown to increase or decrease neurite development. During cell differentiation, neurite development is one of the first steps in showing that these cells are going to differentiate into forming neurons. So I wanted to look at how my mushroom treatments were possibly hindering that development or helping it increase. So our lab had previously shown that the antipsychotic haloperidol did decrease neurite development in a specific set of cells. And so I wanted to compare my mushroom treatment to the antipsychotic haloperidol just to see if those morphological changes were evident between all these groups. So I took PC12 cells, which were from rat adrenal glands. So these are not neurons, and these are just a mammalian cell that can be used for studying neuronal development. So you give them neuronal growth factor and they form these neurite outgrowths, and they do have enough properties that you can do neuroscience studies on these cells after giving them NGF. So I gave them the NGF, let them sit in that for 24 hours, gave them either a treatment of no substances, gave them a mushroom sample that I had extracted in water, or gave them a treatment of haloperidol. After 24 hours of sitting in those samples, I pretty much froze them in time, stopped all their cellular development, plated them on slides, and imaged them. This is what these images end up looking like, and they're definitely hard to see, but that's the experience with microscopy. So they start out as these round cells, and if you're able to see any outgrowths on here where something just doesn't look round, that's exactly what I was measuring. So they do start pretty much looking like circles, and anything that doesn't look like a circle on here is a neurite outgrowth. So I was able to see a cell nucleus and help me count where my cells were on my slides, and then I could trace those outgrowths to quantify the lengths of the outgrowths and then how many outgrowths were occurring per differentiated cell. So my first set of data was looking at just those outgrowth lengths between all my groups. So I had a non-treated group, the mushroom treated group, and I had the haloperidol treated group. I took averages of the neurite outgrowth lengths that I had counted and traced on tracing software between all of those groups and ran a one-way ANOVA and found extremely significant differences between all of my groups. The mushroom-treated groups caused a significant increase in neurite outgrowth lengths, and the haloperidol treatment caused a significant decrease in the neurite outgrowth lengths. This treatment with the haloperidol showing this decrease did validate our previous results. So we're now showing that this experiment can be repeated, and this further validates my own results here on this mushroom sample, showing that the increase that I did have could also be repeated in the future, and that it really was a significant increase that I was noticing here. So this is going to be that first step into looking at these mushrooms may have less of a damaging effect, or they might have more of, it's not necessarily neuroregenerative because we don't know if they're able to repair any damage due to any disorders or medications or treatments at this point. That's a further investigation, but right now it will say that differentiation and growth can occur despite treatment with these mushrooms, and this will even increase that growth. The next thing I looked at was just the amount of outgrowths per neurite. So any cell that I could tell had differentiated, I counted however many neurites were on there, added them to my data, and took an average for each of my groups. I did not find a significant difference after a one-way ANOVA across all the groups. I would like to repeat this one in the NGF for 48 hours rather than 24 hours, just to allow more differentiation to occur before treating the cells with the samples, just to see if more outgrowths are occurring that I can count, and I can have more differentiated cells up front so that if there is a difference, there's a little bit more space in my sample size error so that I can correct from that in the next experiment. So currently what this study implies is that these mushrooms increase the progression formation of neurites. This is the opposite of haloperidol. And this morphology study is also showing that these cells are going to be a good model for doing immunofluorescence studies later on to figure out what proteins are responsible for these changes and what cellular processes are being affected by all of these treatments. You can also then look at co-localizations of proteins involved in synapses to make sure that they are where these outgrowths are occurring, to see if these are neurites that are going to be pruned back later on during development, or if these are functioning and going to be active in communication later on. And a lot of the studies that I ended up doing also were specifically related to looking at disorders that decreased neurite development. One of those included anything that was long-term seizure disorders due to chronic alcohol use or just due to epilepsy, and looking at how these could be used in treatment for such disorders that do not yet have treatments that don't also impair neurite outgrowth. The current treatments for some of these disorders is to give more benzodiazepines and to give more haloperidol, both of which are decreasing outgrowth. So there's not yet something that has the same mechanism of action as the benzodiazepines that is not harmful to development. So the second study that I did was the chemical analysis of the mushrooms. So I took whole mushrooms, I ground them up, and I added them to whichever solvent was going to be important for the study I was doing. I then had to break down the cell walls of these mushrooms. This is a hard part because fungi contain chitin, which is also what makes up the exoskeletons of crustaceans and insects. So I had to break down chitin of fungi to release anything that I needed from the mushrooms, and then just separated out solids and liquids as best I could, filtered and purified those liquids until I realized that it was as clear as possible, and then I had removed even as much protein as possible due to repeated filtration. And then I was able to evaporate any excess solvent and continue to wash the compounds with diethyl ether, which is a solvent that evaporates very rapidly and is extremely lightweight, less dense than all my other solvents, and could pull off any excess solvent from the compounds, and I could end up with an extremely dried result at the end. Those had to be stored as airtight and as cold as possible to prevent any degradation. But I was at that point able to visualize exactly what I was pulling out of the mushrooms and to make sure that it was in the solid state that it should have been according to other experimentation that was similar. And then I would redissolve them for IR visualization, which is a spectroscopic method I ended up using. So infrared spectroscopy is a spectroscopic method that looks at the vibrational frequencies of nuclei interacting. So within a molecule, how are two atoms next to each other interacting with each other? And it passes a light through a sample, which hits a detector, and out onto a recorder. You can see the absorption of that light at certain frequencies. With this, you can see what bonds are in the compounds that you're analyzing and at what orientations those bonds are. After this spectroscopic analysis has been done in a lot of labs with similar compounds, there's been enough evidence to deduce down what functional groups will cause which shapes of peaks across the entire spectrum. And so you can combine all that information together at the end of the day to be able to pull out most of the information of what you have in your sample. This is just some fun images that I had from the summer of me doing IR in the other lab where I had learned to do it for the first time. And I will say IR is usually not a quantitative measure. It was the instrument that was best for my experimentation at this time to collect this data. It's usually used for monitoring reaction progress before you do spectroscopy later on after your reaction to validate your results in a quantitative manner. Instead, I ended up using IR in the entirety of this project just to see as much as I possibly could with the instrumentation that I had available to me. And this data will now propel me into being able to do further spectroscopic analysis in the future. The two psychoactive constituents that I was expecting to see in my mushroom samples were ibotenic acid and mucimol. In these mushrooms, the ibotenic acid is more available, and both of these are neurotoxic, but the ibotenic acid has been attributed to brain lesioning effects. So this is something that you're going to want to avoid in a lot of home remedies that are being created, or any medicine that's being created in an unregulated fashion. Subjective effects to ibotenic acid include a stimulating effect as well as euphoria. Mucimol is less available in the mushroom. However, this is the more neurologically active compound, so it does have a little more bang for its buck, and you don't have to worry about that ratio as much, but there is still a need to reduce the ibotenic acid as well. So the mucimol is less available. It causes dissociative effects as well as that euphoria. And mucimol is also being extracted from the mushrooms in a lot of places in the pharmaceutical industry for studies. There's even looks into cancer treatments at this point, harnessing in that toxicity without having as many negative effects as some of the other cancer treatments. Both of these compounds are specific just to Amanita muscaria, and I think that there are a lot of neuroscience labs that do use mucimol and don't know that this is the mushroom that it's coming from. The only structural difference between these two compounds is that the ibotenic acid has a carboxylic acid on it and the mucimol does not. So the first extraction that I did was I took the entire mushroom and I put it into methanol, and I expected to see mucimol and ibotenic acid come out. So these are your first glimpse at what IR results look like. So typically I will look at what the solvent spectrum looks like. If you've done this enough times, you'll know what a solvent spectrum should look like, but you can compare your solvent spectrum to your sample spectrum and figure out what peaks are specific to your sample versus your solvent. And all of those peaks come in at a specific frequency, and on the y-axis is the absorbance that the recorder is collecting. Both ibotenic acid and mucimol are soluble in methanol. The way that I identified both of these compounds in this methanol sample on IR was I found that there was that OH stretch all the way to the left, which does occur in methanol, but the peak had changed due to my sample. But there's also a shoulder on the left of that peak that I attributed to the primary amine, that NH bond that is on both of my structures as well. There's an NH bond in the aromatic structure as well. There's also the NO stretch, which was extremely specific to my structures. These are called isoxazosyl rings with these NO bonds within this 5-point aromatic ring. So finding that NO stretch really specifically said that I had pulled out the structures that I was looking for. I did see the carbon-carbon aromatic stretches, more evidence towards that primary amine, as well as more evidence towards there being a carboxylic acid in the sample. So at that point now I found carboxylic acid, I found other carbon-oxygen bonds, the primary amine, and the isoxazosyl ring structure, and I now know that in methanol I am pulling out these active products. So I did this again in methanol, and ibotenic acid and mucimol are both minimally soluble in ethanol. When I got back my spectroscopic results, I had no idea what to do with this. And I do also want to say that on all my spectroscopy, I did this multiple times, different concentrations, a handful of different variations, so that I could really use context clues to fill in what I was missing on the peak interpretation data, and anything that I couldn't identify up front, I could identify after taking the spectrum enough times that I knew what was changing due to what, and I could deduce what bonds I was looking at. So in ethanol, everything looked completely different. It was a whole new ball game. Even when I was preparing the samples looked completely different. So that first one I did was the methanol one, which on here is on the right. That one was yellow when I did the extraction, and I felt really confident that I had pulled something out of the mushrooms. I knew that methanol is supposed to be clear, now it's yellow, so I've done something. When I used the ethanol to do the extraction, this came out completely clear, and I felt like this was not going to happen. This was not going to work. There was no yellow showing until I had done enough solvent reduction for that tiny little bit to be showing here. So I knew up front that whatever I was pulling out of the mushrooms was not super soluble in ethanol. And just a fun fact here, when you dissolve these things in water, it's brown because most of the things in the mushrooms are soluble in water. There's a lot of proteins from the mushroom soluble in water. And going back to the cell study, we used what was in water for those cells. So the cells were receiving the full mushroom sample, as much mushroom as they possibly could. So they weren't receiving just isolates of those active compounds. They were receiving mushroom extracts in entirety. But water is not a good solvent for IR. So I was using methanol as a model for water. So back to the ethanol step. I ended up identifying a bunch of carbon-hydrogen bonds. Carbon-hydrogen bonds are really overlooked in IR because just think about it, any organic molecule that you're going to be looking at on IR is going to have carbon-hydrogen bonds. There's no reason to waste time most of the time trying to identify those. But the peaks that I did have, I had no answer for. And after enough time, I was able to identify that there were carbon-hydrogen bonds in here, and they were at different orientations. And there was no reason I should have been seeing this much data for them. If all I was looking at was ibotenic acid and mucimol, it didn't make sense. And I had also lost here some of the structures that were present in the methanol. I've now lost in the ethanol. So I've gained some carbon-hydrogen bonds, and some of the things that I lost here were I lost this primary amine. There's no more shoulder here on this peak. And I also lost this specific bond here that I had mentioned was important to identifying the ring structure. So I don't even have a ring here. And this is a problem because I didn't know any compound. I don't have a ring with the NO in it. I didn't know any compounds that matched that. And then I found this guy. So this is muscarine. Muscarine is one of the psychoactive compounds in Amanita muscaria. If you are doing research on what the compounds are that other people have identified, sometimes I do list muscarine as one of the top 3 products. However, this one's not unique to Amanita muscaria, so I think it did get overlooked a lot of the time. If you look at this thing, it's got a lot of methyls on it. So this fixed that problem of the carbon-hydrogen bonds. It does also have a carbon-oxygen bond. There's 2 different carbon-oxygen bonds in here. So another unexplained peak was resolved. And I needed to, once I had looked up the solubility of muscarine, which is a very commonly used product, so it was not hard to find that information, I found out that it's very soluble in ethanol. So that explained everything, and I felt so much better about this. Other things about muscarine is it acts on the muscarinic acetylcholine receptors. The agonists to those receptors have been used in Alzheimer's disease treatments, and antagonists have been used to treat hallucinations. I do know that a new muscarinic receptor antagonist did just come out for specifically the treatment of schizophrenia, I think like within the past month. So then a lot of the time when you're hearing about mushroom poisoning, it's coming from muscarine poisoning. And once again, it's not just these red ones with the white spots. There are multiple classes of mushrooms that do have muscarine in it, and it's toxic and it's not going to make you feel good. So a lot of the cases that come into the hospital, this is what's responsible. So I wanted to avoid extractions in ethanol because this was not my target compound, but it does give some information of extractions done in vodka, which is common in home remedies, may not be pulling out what people are thinking, and a public health response to this unregulated sale and management of Amanita muscaria is more important than I had initially realized when I began this project. So I went back to looking at the ibotenic acid and mucimol. The whole goal here is to decarboxylate the ibotenic acid to make more mucimol, whether you're in the pharmaceutical industry or just the goal is to decarboxylate ibotenic acid. So there's a couple ways that are hypothesized that you can go about doing this, and very few of them have been validated in any quantitative way. So you can heat extracts, full mushroom extracts, to just below 100 degrees Celsius. Above 100 degrees Celsius, there's suspected that there's a lot of breakdown of the compounds. You can use citric acid or lemon juice to until the pH is between 2 or 3 to cause this decarboxylation. And there was the hypothesis that using vodka would preserve the compounds during the extraction, and there would be a different ratio of ibotenic acid to mucimol than when you used water. But I deserted that hypothesis, and I'll come back to it later in future experimentation, and I wanted to really focus on the citric acid mechanism. No one at this point had identified the method of how citric acid can decarboxylate ibotenic acid. Citric acid's covered in carboxylic acids, so it didn't make sense to add more of them when I'm trying to get rid of the one that I had. So I took the sample, I went back to working with the sample in methanol. That was the easiest one to work with. It really, I could understand what was happening anytime the sample was in methanol. I added citric acid, took a blank sample of citric acid with methanol, and then I combined my mushrooms and the citric acid until the pH was 2.5, had that in methanol, and took a spectrum of that. So with my blank citric acid spectrum and with my mushroom plus citric acid spectrum, I could tell which differences were occurring specifically just to due to citric acid and which were occurring due to my mushrooms. After some mathematical alterations to both of those spectrums, I was able to pull out this adjusted spectrum, and where you could see that increased peaks above that average baseline in there are where things were increasing due to the presence of my mushroom versus citric acid, and decreased peaks were showing where the citric acid structure had potentially changed and some of those bonds had been broken once the mushroom sample was added. So I now knew that this was going to be a great way to figure out what this mechanism was, because I could identify potential reaction intermediates, and I'll walk you through that process of what I ended up finding here. So first I found that my mushroom was indeed in here. So I identified that there was definitely a ring because I have these carbon-carbon aromatic bonds. I have, once again, nitrogen-hydrogen bonds that are showing this amine structure. And I had that NO aromatic symmetric stretch that came from that isoxazosyl ring. The next things that I found were unique was that carboxylic acid stretch had decreased, and citric acid has a lot of carboxylic acids in it, so it should have been extremely high in the first spectrum, and it was. But why did it decrease when I added the mushroom sample, which had a carboxylic acid on it itself? So I wanted to figure out then what is happening to the carboxylic acids on the citric acid that's causing that decrease here. The other really interesting thing is that all the way on the right and up in the top that's appeared there is that NH degradation peak. This is really interesting. There's a peak that's been identified as specific frequency and a specific shape that's extremely clear after a lot of experimentation from other labs, that when a nitrogen-hydrogen bond breaks, this peak forms, or is repeatedly breaking and reforming, this bond forms. So I had that very clear peak on the previous spectrum. It's changed here a little bit just because that solvent subtraction changed some peak locations during this mathematical analysis, but I kind of used all of them together to figure this out. So I now know that the amine is breaking down. But the amine is present in the ibotenic acid and the mucimol. So what part of the mechanism is causing this breakdown, and how is it reforming again during the mucimol's reformation in the last step of the reaction? The last things I found here was I found a nitrogen-carbon double bond, completely unexpected. I have no places where I had expected that to show up, so I determined this to be a reaction intermediate. Another thing is I found that my oxygen-hydrogen stretch had significantly decreased. There's a handful of reasons that this can happen, because some of this is solvent subtraction in this math right here, so it's not as significant as you're thinking based on how this looks normalized to the other peaks on here. However, it is showing that there are some reactions of breaking apart oxygen-hydrogen bonds throughout this entire process, which would be expected because I know that this is going to be acid-base reactions. So this is the mechanism that I developed and I'm proposing for this decarboxylation of ibotenic acid with citric acid. So I first proposed that ibotenic acid is acting as an acid first. So it is being a proton donor, and this then, there's also an assumption on this mechanism that I'm showing here that there's water. This does apply to reactions in methanol as well. I worked it out again in methanol as well, but it was more helpful to have it in this generic water sense, because a lot of this hydronium formation, water formation, and proton removal would be the same in methanol anyways. So after the ibotenic acid acts as an acid, it forms this double bond to the citric acid structure. So this is that first intermediate that I saw appearing on the spectrum with that nitrogen carbon double bond. And this structure now is extremely unstable, so the reaction is going to continue to balance itself out. With that, we'll have the removal of the carboxylic acid as carbon dioxide. So now the ibotenic acid has been successfully decarboxylated. Now the structure is going to act as a base. This is what's called a Schiff base. Now we have two acids that after the beginning of this reaction are acting as a base, and they're going to accept a proton in the next step of the reaction. After proton acceptance, the structure becomes unstable in water or anything polar, and you now have the reformation of citric acid, as well as you'll have mucimol now, so that amine has been reformed down at the bottom there, and there's no more carboxylic acid on the mucimol structure, and citric acid we now discovered is a catalyst in the reaction. So there's still a couple of things here that were not explained on the IR spectrum that we'd still have to keep looking into, but I do know that some of it was solvent reactions that are not explained by this mechanism, just some of the changes to the oxygen-hydrogen bond, some of the changes to where I saw carbon-oxygen bonds. And when I worked this out as a reaction in methanol, I was able to visualize some of those things, but I think further investigations in other solvents, such as doing this in deuterated water and doing NMR, would clear a lot of that up, and I would be able to further validate this as being the mechanism for this reaction. So overall, in the chemical analysis, the full mushroom sample in methanol pulled out ibotenic acid and mucimol. I was using this as a comparison to reactions in water. The best way to compare this again is going to do this in deuterated water and do NMR, or to do mass spec of these compounds as well. But the best way to compare it to water is going to see if we actually put it in water, if the results are similar. The full mushroom sample in ethanol only pulled out muscarine. I had done this because it was comparable to vodka. Once again, this does not contain water in this pure ethanol, and vodka does. So there's going to be some differences here. But the full mushroom sample in methanol with citric acid, I realized the mucimol structure. I did not recognize enough carboxylic acids here to point out that there was a ibotenic acid structure. There should have been some other carbon-oxygen bonds present to show that planar orientation that occurs on the ibotenic acid on the spectrum, and I didn't see that. So there's not an ibotenic acid concentration high enough to be detected by IR in the second methanol sample. And this reaction occurs through a Schiff base intermediate. So the future directions that I want to take this project in is going back to the cell study. I want to do protein quantification studies to see which proteins are responsible for all of the changes that I had observed in cell morphology. Once again, it's to look at how effective those neurite outgrowths are going to be in communication and in true development further on. I want to be able to take my specific isolates now and that I do know the ratios of, further quantify those with NMR first, just to get what the clearer ratios of the ibotenic acid to mucimol to muscarine are, and make some samples that I can give to the cells specifically to see how those compounds individually are affecting cell morphology. I can even follow that up with protein quantification studies, such as immunofluorescence or Western blots. And then I want to repeat the cell study with more treatments. We could add another antipsychotic, don't necessarily have to do that, but there is also a comparison that we can do to the equivalent of vodka. We can do comparisons to benzodiazepines as well at some point. And then I just really want to work on redoing any spectroscopic analysis I can do before I do those things, just so that I know clearly what's in my hands and what these cells are receiving in all of their treatments. So the final implications is that these mushrooms have the opposite effect on neurite growth than does haloperidol. So there's a potential treatment for disorders that have caused a decrease in neurite outgrowth, such as long-term seizure disorders. Home extraction methods involving vodka are ineffective in pulling out mucimol and ibotenic acid. They're instead pulling out muscarine, which may be unexpected by it's just unexpected and can cause harm in that sense. However, I've also now identified this is a good way to pull out muscarine for research that will involve muscarine in the future. And then the last thing is that citric acid is able to decarboxylate ibotenic acid in methanol or water through the formation of a Schiff-base intermediate. So I just want to thank my lab group that was helping me. There's a lot of moral support from that group, as well as my advisors, Dr. Kovacs and Dr. Larimore, and then my lab partner, which was Rebecca Harshman. She was helping with a lot of things this semester. And I will take questions at this time. Thank you. Thank you.
The second one was like how speculative is the reaction, like the molecular reaction from citric acid to something else, to something else, to something else? Is that, are you pretty sure of that, or is that just something that made sense?
Yes. So the reaction that I proposed did model some similar reactions. So it's a lot less speculative than I think I make it out to sound, because that formation of a Schiff-base intermediate in other decarboxylation reactions really follows that way. So the only things that could be incorrect at this point may be just certain electron movements, but those I didn't even put on my mechanism yet because those would be things that I don't quite know yet. Anything that was on there was something that I felt pretty certain about based on I had done, like I said, I had gotten multiple spectroscopy results looking at different concentrations and variations. So being able to see that nitrogen carbon double bond, that was very unique and very unexpected, and I wouldn't have known to look for that. So once I had found that, then I was able to backtrack and figure out what intermediates and decarboxylation reactions have that structure. Once I connected that dot, everything else really clearly fell into place. The dots just kept connecting at that point. So I'm not too concerned about any of that being far from the truth.
Now you say that, could you go back to that reaction?
Yes.
Yeah, so isn't that upper left is the most part of the ibotenic acid? And that's got a nitrogen carbon double bond. Is that not?
Not significantly enough where it was appearing in the methanol spectrum. And although IR is not necessarily used for quantitative analysis, it does show an absorption kind of in a percentage type way where if I did one spectrum and it's showing extremely low and I'm doing another where it's showing extremely high, those can be compared against each other. I think there are some spaces where it does get disregarded and they say, well, we're going to do NMR anyways, so let's just figure it out later. And that is not necessarily what I could do in this situation. And so I just kept getting more data until I could really see that that peak was due to something that was not in the methanol sample.
So on NMR, so this is, if you had NMR that would just nail this down even more.
Yes, NMR is more, it's a more reputable quantitative analysis for looking at things like exactly like this, where I am looking at ratio changes due to specific changes in my reaction conditions. Yes.
In your future research that you were talking about, how are you hypothesizing that the mucimol is what's causing the abnormal, or not abnormal, but the outgrowth that you were talking about, the neurite outgrowth?
Yeah, so at this point I wouldn't know because I gave the cells the entirety of the mushroom sample in water. Remember that was that brown looking one. So there's a lot more in there than just mucimol, including I believe there's a lot of proteins that I hadn't that I did not have in anything I was doing IR on. So they were getting a lot more than just mucimol, and I wouldn't know specific mucimol reactions besides just looking at what other labs have done in similar research. So that's what the point is in the future directions, is to make my isolates that have just mucimol. I mean, you can also buy mucimol, but if you have the skills to extract it, do that. And so I just want to be able to make all of those specific compounds and know exactly what's in there and repeat the cell study so that I can figure out what was responsible for the changes I saw. Anything else? Yes.
For this mechanism, is there any hypothesis about how this same mechanism works in vivo in the mushrooms, or would you be able to apply this to like what's happening in the actual mushrooms when they're growing, or is this mostly just for pure purification?
Yeah, so in the mushroom, I was wondering about that myself because as we know, mushrooms are, most of them are going to be more than 90% water, and this reaction is occurring in water. But what I did find to be the difference was the air exposure. Mushrooms are extremely sensitive to their air exposure, so that's, I believe, why they're going to have this chitin structure that is protecting them, preventing this degradation, because as soon as you put these structures in the air, they decompose into something unrecognizable. There are some studies showing what they decompose into, but they don't align. So there's not yet a way to clearly see what would happen. So I was wondering why, when these mushrooms are harvested, why is it mostly ibotenic acid rather than mucimol? When if you put ibotenic acid out in the air, or if it's ingested and it's in the stomach, it rapidly converts to mucimol. Why does that not happen in a mushroom that's mostly water? And I think it is just due to the cellular structures protecting everything that the mushroom is functioning on. They seem to be so much more complex than you would have thought at the beginning. So, yes. Yes.
Okay, so I'm kind of coming at this from an anthropological religious studies standpoint. So I don't know about chemistry, but this NH degradation peak that you were talking about very much kind of mirrors this this breakthrough, this emotional breakthrough measure in like in cultural studies. It's just like when you come over this peak and you're like coming to all these realizations, you experience fear, terror, you're coming to all these like, and then you kind of come over this crest and you find transcendence, it depends on like if you're using it for spiritual reasons or, personal development reasons, recreational reasons. But there is this breakthrough that happens and then this transcendence effect. And I thought that kind of mirrored the experience. I don't know if the graph kind of I don't know.
That's really funny to hear. I like that. So reading, if you look into the history of the mushrooms and you're looking at, there's a lot of there's books on the spiritual revelations that occur due to consumption of the mushrooms and like what a lot of the folklore that has been developed, how that's happened. And there's a lot of listen and the mushroom will teach you. And while I did not believe that to be true, I didn't know what to do when I started. And by the end, I felt very confident about a lot of the things that I had found out. And I realized that I knew a lot more than I had thought. So this was a great project of being appreciative for the red and white mushrooms that we thought weren't real until, most of us didn't think they were real until we came in this room, if you admit it. So, yes.
Why do the mushrooms produce these compounds? Like, what does it do for the mushroom?
So I have not gotten to look into that. I was wondering about psilocybes as well. I've been wondering about that from an evolutionary standpoint, and so I'm sure someone else has researched it, but I haven't gotten to that point yet. I'm just really interested as to why those guys survive. And they're using it, some of it even doesn't add up from an evolutionary standpoint as well, just looking at natural selection, because reindeer love these things. and this, this is why the folklore of Santa Claus happened, and flying reindeer and this idea. So although there's not direct ties to if those texts were really saying that, it does make sense why the folklore itself happened. But there is a history of these mushrooms being consumed by the reindeers urinating, and since the reindeers have consumed the mushrooms, the ibotenic acid's been converted to mucimol, and so people would drink the urine to get just the mucimol without the negative effects from the ibotenic acid or from any of the structures in the mushroom that couldn't be digested. So yeah, I'm trying to figure out how hallucinogenic mushrooms in general have survived the test of time.
They spore before they're eaten.
Yeah, there is that too. Well, it's not always true though, because they do have their full nutritional value from the time that they're pins too. So, they could get eaten at any point without sporing.
But generally they spore.
Yeah. And a lot of their network that they survive on is what's underground, because what's above ground is just their reproductive organs. Yeah. So what's underground is actually their community and their lives and their bodies. So yeah.
So the psilocybin genes are interesting because it looks like they're horizontally transferred like as a packet across evolutionary time. So they have vertically transferred, not all of them, but it looks like they've been horizontally transferred into different groups, which is interesting.
Yeah, that was going to be one of my investigations.
No one does, no, because fungi are weird and no one knows enough about it. But yeah, so they don't look like they were vertically, like it's not, because you see it across different groups, and it's not that they've independently evolved psilocybin in different groups, but rather it seems that it happened once and then was transferred. Because fungi swap genes.
Yeah. Mm-hmm.
Not as much as bacteria swap genes, but more than we swap genes. More than we care. But no one's really.
That's cool.
It is cool. No one's figured it out.
Do they swap them by hybridization, or do they swap them by the vector?
It might be vector, yeah. But also like fungi connect. Yeah, they do the connection thing, so they might have kind of done it that way. It might be some kind of hybridization.
It is, but I think a lot of them are different, but I do know that hybridization occurs. Like if you have two mycelial networks of two different species growing next to each other, there's a chance that they will eventually merge, and that you can have something in the middle that's both, and things on the outside that are separate. Or you could have things that are separate species still, but the ones that were in the middle having some random traits of the ones of the other one. But I think they're completely different across all. They're all different. So
yeah, but no one's really worked out like how it got into the nuclear DNA. But that's kind of horizontal. That's the story. Horizontal gene transfer. I don't know. It's interesting. It's really cool.
All right. That is all I have. Thank you.